Madeline Potter here. Today we are in the Shrewsbury entomology lab, and today I'm going to go through what my citizen science project is all about, called Project Stink Be Gone 2, and some cool things that I'm finding come out of the insect eggs my citizen scientists are collecting. So the first thing I do when I get to my entomology department building is I go to our packaging office and see if I've received any citizen science packages with this awesome, we have our Project Stink Be Gone 2 stamp on the side, and then I'll bring those down to the Shrewsbury lab. So I open up each one of these boxes and they each have a small soft cooler for the Project Stink Be Gone 2 project. And that's where the insect eggs are kept and preserved for shipment. So inside the soft cooler, my citizen scientists include some packing material so the insect eggs aren't kind of moving around too much and probably damaging our insect eggs. There's an ice pack in there that's covered in paper towel to prevent any condensation from getting on the eggs. And then they'll include any of the petri dishes, and sometimes the insect eggs will be a little too big to fit into a petri dish, so they'll place them in a Ziploc bag, especially our Chinese mantis egg sac or Utheca that we have here. The other thing the citizen scientists include in their package to me is a filled out data sheet. This one is blank just to keep confidentiality of my citizen scientists. But this is where they fill out all the information, the collection information about their insect eggs, and includes them labeling their site numbers that they search, their certain stop time, county that they are searching in Maryland, GPS coordinates, host plant name, and so on. So this is one of the most important parts for me so I can input this data into my own data sheets. So when I'm going through the insect eggs that the citizen scientist sent me, if something is too small to see with the naked eye, or if I want to get a closer look, I'm going to look through my microscope and confirm that what they sent me is or is not an insect egg. If it is an insect egg or egg mass, it will get its own unique egg mass ID code, and then it will be placed into a growth chamber. So what is a growth chamber? Right behind me we have two growth chambers here. This is specifically the one that I'm using for my project right now, and this is where insect eggs and egg masses are going to be placed. Growth chambers are like giant refrigerators, and what's cool about these is that we can control not only the temperature, but we can also control when that temperature may be changing. We could make it colder at night, warmer during the day, and we can also control the timing of the lighting, so we can have a certain daylight cycle for the eggs. And this just helps us mimic the environment outside, and so we can rear out any insects that are inside the eggs as if they were still outside, how my citizen scientists had collected them. And we'll go ahead and take a look inside here. It's a little bit noisy, we can hear the fan going. Right here I do have an extra temperature and humidity gauge just to keep an eye on how things are going within the growth chamber. And we can see here the trace, and all of these little vials in petri dishes are insect eggs or egg masses that the citizen scientists have collected. We are now over 200 eggs or egg masses collected, which is really awesome for being a month and a half into the project. And so we can take a closer look here and see that we have insect eggs inside here. Every 2 to 3 days, either my lab technician or I will check all these insect eggs for any sign of emergence. There could be some caterpillars, some mantids, or hopefully some parasitic wasps or flies that have emerged from these eggs. So an example of checking emergence, we see a large petri dish here and 2 parasitic wasps have emerged. We'll take this petri dish over to the microscope. We see this is a Carolina mantis egg sac, and the 2 parasitic wasps, how big they are in relation to my finger. I will then take out some glass vials, which I will fill with ethanol and place a special label inside with a unique vial code and the corresponding egg mass code from which we will be taking the parasitoids from, and they'll be placed in this vial for preservation. So when I have parasitic wasps or parasitic flies emerging from insect eggs, and we're having those in the petri dishes or maybe a plastic vial, I'm going to transfer those parasitic wasps to a vial of ethanol is using what's called a aspirator. So I have my aspirator here. There's a mouthpiece right there, a flexible plastic tubing part, and then a piece of mesh acting as another filter, and then a hard glass piece at the end. And so how I'll use this aspirator is I'll put my mouth on this part, I'll suck in some air to suck up a parasitic wasp, maneuvering this end around that parasitic wasp. It'll suck up into this tube, it'll stop right at this filter portion, and then I'll be able to put my finger over this end to trap that parasitic wasp or fly. And then when I'm ready, I'll place this end over a vial of ethanol and blow that parasitoid out into the ethanol vial. And this is to preserve the specimen so we can ID it to genus or species later. Let's go ahead and take a look under the microscope and see some pretty cool things that I've found emerging from insect eggs. The first couple of videos and photos are of parasitic wasps that have emerged from insect eggs. We have the species Anastatis reduvii, and we can see the top one is a larger female, and on the bottom is a smaller Anastatis species male. We can see how small these parasitic wasps can be, especially these guys. These are fairy wasps in the family Mimiridae, and these came from bark lice eggs, as we can see 2 of them on top of this egg mass. And this is a fairy wasp in comparison to the end of a small paintbrush. We're now going to look at some nymphs, and we have some wheel bug nymphs here on top of their egg mass. And the yellow one has just emerged. They're lightly colored, and this is what they look like when they first come out of their elongated small egg. So in order to fit inside, they're folded up, and they'll eventually begin to unfold their legs and their antenna. Once the nymph has unfolded itself, within a couple hours it will begin to darken into this black and red coloration, and you can also see on their antenna are these bright yellow tips. With this nymph walking around, you can also see its rostrum, which is their piercing, sucking mouth part. It's a straw-like mouth part that they use to stab their prey and to suck on bug juices. And you can see it's almost like a long nose coming out of the front of its face that it's moving around. When we zoom in on wheelbug egg masses, we may see some other creatures. We have a mite here, which is an arthropod, walking around. Some other creatures that make homes out of egg masses are these springtails in the order Columbella, and they were found crawling around some mantis egg sacs. I also found this true bug in the order Hemiptera nymph walking out of a mantis egg sac, and it's likely either a plant hopper or a tree hopper nymph. These are some katydid nymphs coming out of their eggs. You can see when we zoom in on one that is slowly moving and wiggling its way out of its egg. These tiny insects are bark lice nymphs. These are caterpillars in the family Noctuidae, which is a moth family. Here we have some Chinese mantids freshly emerging from their ootheca. If we zoom in, we can see 2 little nymphs beginning to wiggle or worm their way out of their ootheca. And just like the wheelbug nymphs, they'll begin to unfold their limbs once they find their way out. Eventually, they'll begin to darken, going from a yellow color to a brownish gray color. Now, if we have insects that emerge that are not parasitoids, they're not a parasitic wasp or parasitic fly, like we have here, we have some baby or nymphal mantids. These are Chinese mantids, and a lot of them can come out of this egg sac, which is called an ootheca. And so when I have these, there's no parasitoids flying around in my petri dish. I'm gonna take it outside and I'm gonna release all these baby mantids so they can do their amazing service as being predators out in our landscapes and eating those pest insects that feed on our plants. I'm gonna take this egg sac back to the lab within this petri dish, place it back in the growth chamber, and to see if any parasitoids will still come out. Thank you for watching and following me around the Shrewsbury lab to see what is going on with Project Stink Be Gone 2. I would like to give a huge thanks and a huge shout out to all my citizen scientist volunteers. I could not do this project without them. A huge thanks to my Shrewsbury lab family, especially my PI, Dr. Paula Shrewsbury, my lab technician Adelaide, and my lab manager Nancy. Thank you for watching. Have a wonderful rest of your day and stay curious. I'll see you next time. You can also stay connected to Project Stink Be Gone 2 by checking out the project webpage or Instagram, both linked below.